Welcome to this explainer. Today, we're diving deep into the fascinating molecule-to-medicine journey of eicosanoids. We'll be mapping out their profound impact on human health, sticking strictly to the science from our source literature. If you're working in the life sciences, healthcare, or honestly, if you're just deeply curious about human biochemistry, you already know that the body's microscopic control systems are where the real magic happens. So today, we're zooming in on a class of molecules absolutely central to that control. Let's get started.
Here is our roadmap for this explainer. First, an introduction to eicosanoids. Then we'll hit biosynthesis and pathways, followed by physiology and catabolism. After that, we'll look at pharmacology and disease, shift over to diet and modulation, and finally wrap up with their overall biomedical relevance.
All right, Part 1. Introduction to eicosanoids, the local messengers.
So what exactly is an eicosanoid? Well, the name itself gives us a huge clue. Eicosa literally means 20. These are incredibly potent, short-lived biological mediators derived from 20-carbon polyunsaturated fatty acids, or PUFAs, which are actually found stored right inside our cell membrane phospholipids. Now, in most species, humans absolutely included, arachidonic acid is by far the most dominant precursor. So whenever we're talking about eicosanoids today, we're mostly talking about the direct derivatives of arachidonic acid.
Now, you're probably used to thinking about hormones as these long-distance travelers, right? Systemic hormones are produced in a specific gland, dumped into the bloodstream, and travel all the way across the body to hit their target organs. Eicosanoids are totally different. They act locally. They're synthesized right on the spot, on demand, in the exact cells or the immediate microenvironment where they're needed. They bind to local receptors, do their job, and then quickly vanish due to rapid inactivation. They are, quite literally, the ultimate local messengers.
Moving right along to Section 2, biosynthesis and metabolic pathways, generating the messengers.
We've got a whole family of these molecules: prostaglandins, thromboxanes, prostacyclins, leukotrienes, and lipoxins. If we just look at the prostaglandins for a second, they all share this 20-carbon skeleton based on hypothetical prostanoic acid. The naming convention is actually really intuitive once you know it. The specific letter designations, like E, F, or D, tell us the exact chemical substitutions on their 5-membered cyclopane rings. Meanwhile, the numbers, like the 2 in prostaglandin E2, tell us exactly how many double bonds are in the side chains.
But how do we actually go from a dormant cell membrane to an active messenger? Well, the essential first step is releasing that precursor fatty acid. When a cell membrane gets disturbed, it activates phospholipase A2, a membrane-bound enzyme. This is the absolute rate-limiting step. Once that free arachidonic acid is finally liberated, it basically hits a fork in the road. It can go down the cyclooxygenase or COX pathway to create prostaglandins, prostacyclins, and thromboxanes, or it can veer down the lipoxygenase pathway to generate leukotrienes and lipoxins.
But wait, there is a third, super fascinating route, the cytochrome P450 pathway. This enzyme system can convert arachidonic acid into epoxy-eicosanoic acids, we'll just call them EETs, and HETs. The crucial takeaway for this specific pathway is that unlike the COX and lipooxygenase products, which are strictly made on demand, products like EETs can actually be found stored directly in cellular phospholipids. This hints at a much deeper, sustained role in cell membrane function that biomedical researchers are still actively trying to unravel today.
Let's jump into Section 3, Physiological Roles in Catabolism, Maintaining Systemic Homeostasis. Honestly, it's almost impossible to overstate just how biologically vital these molecules are. I mean, they are everywhere. In reproduction, prostaglandins like PGE2 and PGF2-alpha are absolutely critical for ovulation and inducing labor. Down in the gut, PG-series prostaglandins inhibit gastric acid, literally protecting your stomach lining. In the brain, they're the ones mediating your fever response when you get an infection. And in the immune system, leukotrienes like LTB4 act as these powerful homing beacons, pulling neutrophils right to the sites of injury. They are the microscopic operators keeping our entire system in homeostasis. A really perfect example of their localized balancing act happens right inside our blood vessels. Think of it as a constant tug of war. Inside your platelets, an enzyme called thromboxane synthetase produces thromboxane A2. This is a highly potent vasoconstrictor that strongly stimulates platelet aggregation. Basically, it promotes clotting. But conversely, the cells lining your blood vessels, the vascular endothelium, produce prostacyclin, or PGI2, which does the exact opposite. It's a powerful vasodilator and it actively prevents clots.
Your everyday vascular health entirely relies on this constant microscopic tug-of-war between these two local eicosanoids. Now, because these are local messengers, they have to disappear quickly. If they just circulated endlessly, our cardiovascular systems would descend into total chaos. So it's vital to remember that organs like the lungs, liver, and kidneys are designed to rapidly metabolize and completely inactivate these compounds. Get this. Eicosanoids infused directly into the blood lose their biological activity after just a single passage through the lungs. That rapid catabolism is what guarantees such tight, localized control. Which brings us to Section 4, Pharmacology and Chronic Diseases.
This is where things get incredibly practical. Understanding these specific isoforms explains exactly why nonsteroidal anti-inflammatory drugs, NSAIDs, work, and why they have side effects. You see, COX-1 is constitutive. It's the physiological housekeeper that protects your stomach lining and kidneys. COX-2, on the other hand, is inducible. It rapidly ramps up during inflammation to drive pain and swelling. Traditional NSAIDs blindly block both, which cures the pain, but can cause gastric ulcers. This realization drove pharma to develop selective COX-2 inhibitors to target just the inflammation, though as we know, some of those later presented cardiovascular risks. And fascinatingly, a variant called COX-3 seems implicated in pain and fever, but not inflammation, which actually makes it the primary target for acetaminophen.
Let's look at one of the most widely used medical interventions on the planet, low-dose aspirin for cardiovascular protection. Its success relies entirely on this biochemical mechanism. Aspirin is totally unique because it irreversibly alters the COX enzyme. Now, because blood platelets literally don't have a nucleus, they can't synthesize new enzymes. This means aspirin permanently cripples a platelet's ability to make that pro-clotting thromboxane A2 for its entire 8 to 10-day lifespan. Meanwhile, the cells of the vascular endothelium do have nuclei, so they just rapidly synthesize new COX enzymes and go right back to producing the protective, anti-clotting prostacyclin. It's a brilliant biological hack.
But it's not just about heart health. Eicosanoids are heavily implicated in cancer, too. The literature clearly notes an overabundance of COX-2 in colorectal, prostate, breast, and lung tumors. And it makes sense when you look at the mechanism. Excessive COX-2 actively promotes tumor angiogenesis, meaning it builds the new blood vessels that feed the tumor, while simultaneously inhibiting apoptosis, which is the natural programmed cell death that's supposed to clear out those defective cells. Understanding this mechanism helps explain why massive population studies have actually shown that long-term NSAID use is associated with a significantly decreased risk of certain cancers.
All right, Section 5, Diet and Eicosanoid Modulation. We're moving beyond pharmaceuticals now because our daily nutritional choices fundamentally dictate what our bodies can even synthesize in the first place. Specific dietary constituents directly interact with our synthetic enzymes. For instance, vitamin E can inhibit phospholipase A2. Alcohol can inhibit thromboxane synthetase and mess with desaturase enzymes. Even vitamin C acts as an inhibitor at certain concentrations. So our diet isn't just fuel, it actually provides direct chemical modulators that profoundly alter our entire lipid mediator profile.
And this brings us to a massive concept in nutritional science, the omega-6 versus omega-3 balance. A standard Western diet is packed with omega-6, yielding abundant arachidonic acid, which, as we know, produces highly inflammatory leukotrienes and pro-clotting thromboxanes. But if you consume marine omega-3s, like EPA, they actively displace arachidonic acid right out of your cell membranes. When COX and lipooxygenase are forced to act on EPA instead, they produce variations like thromboxane A3 and leukotriene B5. These are drastically less inflammatory and way less prothrombotic. By simply changing your dietary fats, you are fundamentally shifting your physiological baseline toward a much less inflammatory state.
And it's not just marine oils. Alternative medicine taps directly into these biochemical pathways too. Have you ever wondered why epidemiological data so often points to lower cardiovascular risks for people who eat a ton of garlic and onions? It's because they contain compounds that actively block both thromboxane synthesis and the 5-lipoxygenase pathway. Similarly, curcumin, the active compound in turmeric, is a heavily studied inhibitor of both COX-2 and the inflammatory transcription factor NF-kappa B, actively suppressing the exact genes that mediate joint destruction in inflammatory conditions like arthritis.
Finally, Section 6, Conclusion, the biomedical relevance of eicosanoids. Which leaves us with a truly compelling question. We've just mapped how arachidonic acid drives inflammation, how a simple aspirin intercepts clotting, and how marine oils can literally redefine our cellular membranes. As researchers continue to map the delicate balance of these pathways and dive deeper into localized regulators like those cytochrome P450 products, how will future nutritional and pharmacological interventions further harness eicosanoids to prevent or even reverse chronic disease? The answers are out there, hiding in the continuing exploration of these fascinating local messengers. Thank you so much for joining me in this explainer, and keep exploring the science.